Last speaker, Dr. Shaimaa, Swiss Canal University. The title is very big title. ربنا يسهل والموضوع يبقى سهل. Association of vitamin D and retinoid X receptor expression and vitamin D binding protein variant with bone density in thalassemic children.
Good evening, everybody. First of all, I would like to thank Professor Dr. Amal for inviting me today. Thanks to my chairperson, my dear professors and colleagues. I'm going to cut it as much as I can short, so don't worry. I'm going to talk about vitamin D in thalassemia. So I would brief a very small background before concluding what we have found in our results. Osteopathy is common in thalassemia. It's multifactorial and might occur as a consequence of the disease itself. For untreated patients, osteoporosis, cross failure, delayed bone age could occur. Even for well-transfused and well-chelated children, osteopenia could occur. Vitamin D deficiency is prevalent among children and adolescents with thalassemia. It was found in multiple researches to be 73%, 80%, and 90% in US, North India, and Thailand. Vitamin D is essential, of course, for the calcium homeostasis and bone mineralization, as we all know. So what is vitamin D receptor gene? It is the most biologically active receptor. It has a high affinity to form some sort of nuclear transcription factor to regulate the synthesis of protein involved in bone mineral homeostasis and cell proliferation. The vitamin D receptor gene is located on chromosome 12. It contains many single nucleotide polymorphisms that could potentially modify the expression of the vitamin D receptor. Many polymorphisms has been studied before. The most abundantly found in the literature is the FOC1 and the PSM1 polymorphisms. This study was conducted in Peniswave. They studied the vitamin D level in thalassemia children compared to the control, and they found it to be lower. And when they tested the vitamin D receptor FOC1 and PSM1 gene polymorphism, they found that the patients harboring mutant capital F small and FF small and wild BB genotypes were associated with lower serum calcium in that respective. This is the results. We can see that the vitamin D level is lower in the mutant type in the upper part of the slide, and the PSM1 gene, the wild type, had lower vitamin D levels. This is another study conducted in Cairo, searching for the predictors of bone diseases in Egyptian prepubertal children in beta thalassemia, and they found that there are reduced bone marrow density more in male patients with genotype PP small and FF small, but not to females. The retinoid X receptors. The vitamin D receptors must form a heterodimer with the retinoid X receptor before it can exert any effect in the cells. The retinoid X receptor, like the vitamin D receptor, is a ligand-activated transcription factor that plays a role in the cell differentiation, cell growth, apoptosis, and it also required for proper vertebrate embryonic development. In contrast to the vitamin D receptor, study is not very much enormous regarding the association between the retinoid X receptor gene and other health outcomes, but West et al. Found that there is a significant association between the RS3132299 and the 125 hydroxyvitamin D. The vitamin D binding protein is a GC globulin, is the gene for vitamin D binding protein regulation, and it was assigned to human chromosome 4q9. Genetic variability of the GC gene was found to affect both the circulating vitamin D binding protein and the 25 hydroxy level. And a school-based survey reported that children and adolescents in Finland defined that the GC genotype has independent determinant of bone mineral content in both gender, as well as an association with vitamin D and parathyroid hormone concentration. There are different genotypes for the binding protein, differs by race, and the difference in the race affects the vitamin D level at the end, and of course the concentration of the vitamin D itself. This is another study testing the association between the polymorphic variation in the vitamin D receptors and retinoid X receptor and the circulating level of vitamin D metabolites. This study assumed that many studies found results about the association between the vitamin D receptors and the other genes and the health outcome at the end, but not the circulating metabolite. So when they tested it, they found that the genetic variation in the retinoid X receptor may influence the 1.25 hydroxy vitamin D homeostasis, but it doesn't affect the 25 hydroxy one. This is the result. And here we go about what we have found in South Canal area. Based on this whole background, we conducted a study to evaluate the vitamin D status in thalassemia children in relation to the vitamin D receptor, the retinoid X receptor, and the expression of the vitamin D binding protein polymorphism. This study was conducted on 44 thalassemia children compared to 40 healthy ones. Bone chemistry profile, 25 hydroxy vitamin Z, DEXA, and single nucleotide polymorphism and gene expression was conducted. And surprisingly, that was our result. Vitamin D level was higher in thalassemia children rather than the control, significantly higher, it's around 41 versus only 18 in the controlled one. And of course, the amount of the children having sufficient vitamin D level was higher in thalassemia children, and the calcium level was higher in thalassemia. That's what I was mentioned. Thalassemia children compared to controlled had higher level of calcium and vitamin D. We mentioned this already. So, we found that both vitamin D receptor and retinoid X receptor levels are markedly more expressed in thalassemia children when compared to control group. And it is also more expressed in thalassemia major more than those with thalassemia intermediate. Before skipping this point, besides the higher expression of the vitamin D receptor that we found in the South Canal area, most of our study group, the thalassemia children, all of them is regularly taking vitamin D and calcium, the RDA supplementations they need, but only 40% of the control ones received their vitamin D and calcium, so this is some limitation we faced. And regarding the vitamin D binding protein genotype, the genotype of vitamin D binding protein, the RS4701 and the RS4588 revealed no significant difference between the patients and the control, so it's not about the vitamin D binding protein. But the TG genotype showed high frequency of osteoporosis among thalassemia patients, while both homozygous states, T capital and GG, were common among osteopenic patients. However, there was no founded association between the vitamin D binding protein polymorphism and any of the laboratory and the molecular data. This is the summary of the associations we tested. And in conclusion, thalassemia children had higher expression levels of serum vitamin D receptor and retinoid X receptor, and the vitamin D binding protein polymorphism were associated with bone osteoporosis in thalassemia children. Thank you very much.
شكرا دكتورة شيماء لالتزامك في الميعاد يعني.